Fantastic to be here, and I've never actually been to this part of the country before, so as I drove over yesterday I really enjoyed, beautiful sunshine and it's spring. And spring is very special to me because what the lab has focused on since I arrived back from California as a postdoc is flowering time and how plants use seasonal cues to control their developmental transitions. So it's a topic near and dear to my heart, how plants actually monitor these different environmental cues and then use them to make these developmental decisions. And people now, it's turned into a very interesting area, and people say, gosh, how did you decide what to work on? Because as a postdoc, of course, it's always the biggest challenge. What am I going to do when I set my own lab up? And seasons, because I grew up in the Northwest and then went to university in New York, they were just something that happened in winter, all the leaves fell off and everything, and it was just sort of happened when you just didn't really pay much attention to it. And then I went to California as a postdoc, and of course seasons are very different there, so I began to notice them. And then what cemented my interest was I bought some tulips from a nursery garden in California, and the man said to me, now put them in the fridge for 6 weeks before you plant them. And that one comment, I think, really has changed my life, because I thought, wow, this is really weird. They not only respond to them, but they need these seasonal cues, these different environmental signals. And so when I had the opportunity to set up the lab, that's what I decided to work on. How do plants monitor these long-term seasonal cues and use that to time their development? Now, at the time, molecular biology, this was in 1988, molecular biology was going strong, but really there was very little molecular genetic analysis to dissect something as complex as flowering time regulation in plants. And it was because of the sort of era of Arabidopsis, which was just starting at that point, that we could actually dissect a trait as complex as flowering time. We knew lots of different plants needed different cues. Some needed long days, some needed short days, some needed cold, desert plants needed water. And if you read the literature, clearly this physiological complexity everybody assumed would be underpinned by equal molecular complexity. And I think for the first 5 years or so of a lot of different labs' efforts all focused on trying to understand the molecular control of flowering time, using the genetic approach and then cloning the genes because of the power of the combination of all these labs using this one system in Arabidopsis thaliana, we actually got to a rather simple view of how flowering time is controlled that's actually relevant for all plants. And I just want to sort of reinforce just how important that adoption of the reference model has been to plant biology as a whole by looking at the most simple way I can describe our current understanding of flowering time control. So basically, this is a schematic. I could have shown you a really scary picture of 400 genes and also with various arrows all over the place. But this is distilled down to give you the same information. Basically, there are a set of genes, floral pathway integrated genes, that need to be switched on for the meristem to then switch to a floral fate. There are many hundreds of those, and they all need to be synchronized in terms of switched on together. And the sort of simplicity is that the different inputs by these different environmental and endogenous cues all converge to regulate that same set of genes. And so it's the quantitative activation of those genes that's important. Now, some of those pathways promote the expression of those genes, and other pathways repress them. So there's this sort of yin-yang going on, where the plant is integrating different signals, and through promotion and repression, at some point, enough of these are switched on that the meristem switches fate. And actually, I think you can look at flowering time in all plants now with this sort of framework and investigate flowering time control in a lot of different species with a lot of different physiological behaviors in a much faster way because of this information. Anyway, so that was probably, took up the first 10 years of when I arrived back in England, which, it was a long time ago. But because of my tulip moment, what we focused on were these pathways down here. How is it that winter is required, and how does winter regulate the transition to flowering? So that's what we focused on. Vernalization using Arabidopsis thaliana. We asked, why do some plants need to overwinter before they flower? How does this process of vernalization prolong cold? How does it accelerate flowering in some plants? And winter, of course, is different depending where you're growing. So how do different plants adapt to different winters? And again, and I'm sure Penny will have seen many of these proposals, we've assumed at that point, like many developmental and environmental signals, there would be a sensor for the cold. There would be a signal transduction pathway of some sort, perhaps switching on a set of genes that were required to activate these floral pathway integrated genes. Classic signal perception, signal transduction. But that's actually not what we found. All these different routes, and this is what I'm going to try and at a high level get over today, all these different routes that we started from could have taken us off in many different places, but actually it's all converged down to understanding the regulation of one target at the level of chromatin. And so non-coding RNA and chromatin regulation turns out to be at the heart of, do the plants need cold? How do they perceive cold? And how do they adapt to different winters? Okay, so I'm going to start off with the first question we asked. Why do some plant species need cold and others don't in order to flower? Why do they have this vernalization requirement? And of course, if you have, as an annual plant, a vernalization requirement, you have a winter annual habit. You germinate, you overwinter, and you flower in spring. And winter annual habits are very adaptive, have a higher fitness in some environments, whereas others, summer annual habit is the preferred habit, reproductively more successful, various reasons, often with very, very cold winters, for example, summer annual habit's better. You can potentially get 2 generations a year with a summer annual habit. Winter annuals tend to be where you have very dry summers. So there are lots of reasons, lots of ecological interesting reasons about which reproductive strategy is chosen. Of course, it's a very important question for many crop plants. Most of our major crop plants have winter and spring sown varieties so that geographical range can be extended for winter wheat, spring wheat, a lot of the brassicas. Fortunately for us, there was similar variation in Arabidopsis. And most people think of Arabidopsis as the plant on the right here. Sort of why we use it is because it cycles rapidly. We can do our genetics fast. But if you take 1,000 accessions from the wild, probably 70% of them or so will have a winter annual habit if you put them in your nice, warm, long day chamber. And that if you vernalize these as young seedlings, they will then look like this. So they have a classic winter annual habit, but there are a lot that are also summer annual. Now, again, I'm going to just give you a sort of high-level overview. When we came down to try and understand what was going on, we bumped into this gene that you'll hear a lot about in the talk. It's a gene called FLC, flowering locus C. As someone said, actually, you should rename it flowering lock, because that's a better description of what it does. So think of it as a lock. I think of it as a break. If you express FLC, it's a Madsbox transcription factor. It represses all the genes required for the meristem to switch to a floral fate. So if you have FLC on, you stay vegetative. Hence the schematic here. And what coal does is switch FLC off. So in a nutshell, to be a winter annual, you make very high levels of FLC in the autumn, and then you wait until the cold switches FLC off. Now, when we looked at the winter annuals, in fact, if you cross winter annual and summer annual types together, surprisingly, you can actually map that winter annual habit pretty much as a monogenic trait. It is a QTL, but there's one major determinant of flowering of the winter annual habit, and that's a gene with a trendy little name called frigida, labeled by a German professor in 1955. I unfortunately adopted these very interesting names that we have to live with. But so frigida was the locus conferring the winter annual habit. And the way we understand frigida is that basically if this is a brake, and I use the analogy of a car, if you've got your foot hard on the brake, it doesn't matter how many of these promotive pathways, the photoperiod, the long days, the endogenous cues, all these things might be trying to accelerate you. But if you have your foot on the brake, you won't flower. And what frigida does is slam the foot on the brake. FLC is the brake, frigida is then the foot. It pushes hard on the brake, it makes very high levels of FLC, and the plants stay vegetative. And what was very interesting early on was as soon as we began to look at the summer annual rapid cycling types of Arabidopsis, we found They had a broken frigida. Frigida was lost. And in fact, just to show you one of the first early slides, this is the frigida gene. It's a relatively simple gene. It's one of those where you look at the open reading frame, it's a coil protein, but it doesn't really tell you very much about the biochemical activity. Our current understanding is it's required for transcriptional activation of FLC, but not in the classic transcriptional activator sense. It's doing something where it's a co-transcriptional effect on the beginning of the transcript involving capping, very complicated. Still got a lot to work out what the mechanistic function is. But if we just look at how frigida is now in the summer annual types, this is our Columbia, classic, everyone uses the Columbia accession. We had a 16-base deletion in Columbia, which shifted the frame of the protein. In Landsberg, there was a bigger deletion that took out the beginning of the open reading frame. And so that was a real surprise. That said that these summer annual types had evolved independently from a winter annual progenitor. Here I showed at least twice, and we've now gone on to sequence a lot of frigidas, and I think we've got 28 totally independent loss of function mutations where winter annual has evolved into summer annual. So somehow, despite the fact if you mutate Arabidopsis, there are hundreds and hundreds of genes which, you know, the mutant phenotype has a flowering time effect. Evolution seems to have picked the same gene over and over again for this winter annual to summer annual evolutionary step. Okay, so that was actually relatively simple, and I think there's some lessons to learn that when you really get to a detailed understanding, things are actually relatively simple. So then we turn to, well, now we know why some winter annual and summer annual plants. How does a winter annual actually perceive the cold and switch off FLC? Remember, as I said, FLC is very high because of frigida, and what happens during winter is it's switched off. Now, this is a very interesting process, vernalization, because it's a quantitative process. The more cold you give the plant, the more the gene is switched off. This is just a northern blot looking at RNA. Very high levels with no cold, 4 weeks, still a little bit left. If you give it 8 weeks, it'll completely be switched off. So that was one interesting thing. Why is it so slow, and why is it quantitative? In some respects, that makes sense, because vernalization needs to be slow and quantitative. The plants need to make sure that they've perceived the whole of winter and not just a really bad week in autumn. So, you know, evolutionarily it makes sense, but mechanistically it was interesting. And the other thing that was interesting is if you take plants out at different times, wherever the repression has got to, it then stays at that level through the rest of development. It's epigenetically fixed at that certain level. So what's the mechanism of that? Now I'm going to build on this. This is basically a schematic of what I've just said, of the FLC expression over the lifetime of the life cycle of the plant. So in autumn, FLC expression is very high. Here's our gene. Red is the sense transcript. The plants begin to perceive the cold, and green now shows this slow switch off of FLC expression, which is then maintained epigenetically at this very low state through spring and summer. And that's when the photoperiod, the ambient temperature, a lot of the promotive pathways are important for actually now switching the plant to the floral fate. But of course, every generation of plants needs to be vernalized, so at some point FLC needs to be switched back on again. In fact, that happens in the embryos. So you switch on, and then you're back at the beginning. So I'm going to build on this schematic as I go through. So we did it in the classic way. We said, let's find mutants that don't vernalize so well, and then try and understand what the gene functions that were mutated are. Started from a progenitor which needed vernalization, vernalized the M2, because most of them were early, and then we looked for the ones that didn't flower so early, had a defective vernalization. And that took us into the world of chromatin regulation. It's always interesting with mutants. You never quite know where you're going to end up. And one of the first, in fact, the second one, Vern2, we cloned these Vern mutants, turned out to be a component of something called polycomb repressive complex 2, PRC2. Now, this is a hugely conserved chromatin complex, all the way from humans, all the animals, all the plants, down to, I forget which of the lower plants it comes out. It's not in Cereviceae, but it's a very widely conserved chromatin regulator. And I'll tell you a little bit about that. Plus, there were two other mutations that I'll talk about in two genes. In fact, the Ambasino lab cloned this one, VIN3, and we cloned the homolog VIN5. Now, PhD proteins, plant homeodomains. These are proteins found in all organisms, despite their name, plant homeodomain. They are really very associated with chromatin regulation because the PhD domain seems to bind histones and modified histones. So they are very frequent components of chromatin complexes. And one of them was cold-induced, which was interesting given that's what the process was going to be induced by. Now I'm just going to give a little primer on chromatin regulation because you get into this, the jargon gets quite complicated. So if we unwrap our chromosome, of course what we've got, if we unwrap it far enough, it is the DNA double helix. But the DNA is wrapped around these nucleosomes compacted in the cell. And the nucleosomes are made, this is my artistic version of a nucleosome here, the DNA wrapped around, are made up of core histone proteins, 1 to 4, and there's 2 of each in those. And they have tails that stick out of the histones. And those tails are post-translationally modified. And those post-translational modifications make a huge effect on whether that gene is expressed or not. So that's part of the epigenetic regulation. And of course, you'll have also heard of DNA methylation. This is where the chemical modification is directly on the DNA itself. So DNA methylation and histone modifications are hugely important for gene regulation. If you don't have to look very far to see quite a lot of stuff in the newspapers, transgenerational effects on epigenetics by, smoking mothers and what they're doing to their babies and their grandchildren. There's a lot of press about epigenetics at the moment. People see that the DNA is the sort of hardware in a computer analogy, and then what the writing on top of the DNA or the chromatin are these epigenetic marks. So that's the good way of thinking about it. It's the instructions for how the genes are to be regulated. And so what we'd ended up cloning out on our polycomb system was actually an enzyme that delivered one of these post-translational modifications. Now again, the names are horrible. So histone 3, lysine 27 trimethylation. There's one of them. And so then there's another one, lysine 36 trimethylation. And so these numbers become a little hard to remember. So I'm going to do them in a color coding. So the active modifications are going to be color coded green in a traffic light system, just so you don't have to remember all the numbers. And the silent ones red. So the polycomb is delivering this K27 trimethylation, so our red off state. So we had a mutant. It was apparently involved in delivering these histone modifications, which was somehow involved in the epigenetic silencing of FLC by cold. So then we looked at FLC itself, at this particular modification. And I'm not going to show very many of these pictures, but I just wanted to show one of them. So remember, this is our red modification on our nucleosome here. This is across the body of the gene. And what was very interesting was that where we saw the modification was not actually at the promoter. It was internal to the gene, actually at the beginning of the first intron. This is the schematic. These are little exons. There's 7 exons in FLC, and a very large first intron that's got a lot of regulatory elements. So this important nucleation region, as we call it, which is the important step in the initial silencing, involves accumulation of this silent mark at this site. And as you give increasing cold, so 2, 4, 6, and 8, you increase the level of that modification at that site. So it very nicely is quantitative, which explained in one way the quantitative silencing with cold. Now, the way I look at this nucleation is it's a bit like a zip on a jacket. This is the hard bit to do. When you actually get the 2 ends and you kind of fiddle around and you've got to get it together. That is what nucleation is in my view. Then what happens is that, and I'm not going to show all this, but the chromatin machinery then spreads across the body of the gene, and it's like the zipper going up. And that then holds the epigenetic memory through multiple cell divisions. Now, but the one thing I do want to talk about is this quantitativeness, because when we'd first seen that quantitative change, it's like, okay, well, now we've explained the quantitative silencing. We have quantitative accumulation of the histone modification. But actually, when you really start to think about how is that modification inherited from one cell through the next cell through the next cell, how is 20% of the level inherited versus 50% versus 70%? That's quite hard to imagine, a very robust mechanism that can transfer sort of quantitative levels. And it was then we began to realize that there was an alternative explanation to this nice quantitative silencing that we see, in that it wasn't quantitatively going off in each cell, but what was happening was that actually it was a switch, so you were either on or off, and what was happening was that the proportion of cells being switched off was increasing with the cold. So I just draw that in boxes here and pretend these are little cells in the plant. So blue would be on, white would be off. And you can see as if you grind those up, it would give you that nice picture that we were sort of so used to seeing. Well, so now I'm going to do it in a slightly more realistic scenario of a root. Now people often ask me, Caroline, you know, you're working on flowering. Why are you always imaging roots? Fortunately for us, actually vernalization is a whole plant process. And in fact, lots of the signals, the mobile FT signal is produced all over the plant. It's just that the shoot apical meristem responds to those, which means that you have to vernalize throughout the plant, which means we can look at FLC regulation in the root. Much easier. Anyway, so this is the root, and I'm going to show some pictures, but I just want to show schematically what I'm going to be showing you. So here's a root tip, the root meristem. Green is on, so here our cells are all expressing FLC. Red is off. And what happens with increasing cold, and of course there's not much growth when the plant is in the cold, but we've found that there's a perception, and it's a digital perception of the cold. The cell suddenly decides, I have had enough cold, and registers that cold by nucleation of that polycomb silencing at that internal site in the gene. And so FLC in that cell, and in this case it's 2 cells, switches off. So now you're red. And so as you increase the weeks of cold, more and more of these cells have registered that they've had cold and switches the gene off on a cell basis. Now why I only draw, this happens in all the cells, but why I just draw the meristems here is that they will then provide the bulk of the cells as the plants come out and grow again in the warm. So when you bring the plants back out in the warm, of course they merrily grow away, the roots take off, and so what happens is that these cells in the meristem now divide and produce files, daughter files through the anticlinal divisions, and whatever state they were in the cold is then remembered through the subsequent cell divisions. So you have these files of cells now where the gene is switched off, red. And with increasing cold, you get more and more files of cells that have switched off to the point where now it's all off. So this is what I'm going to show you. So what I'm going to show you is now FLC GFP, in fact Venus. So it's not nicely green and red, it's Venus and dark, but hopefully you'll get the idea. So this is now a real root expressing FLC Venus with very little cold, and you see all the files expressing FLC Venus in the nucleus. But as you go on, you can see now the files of cells are beginning to switch FLC off. Here's one which is only 1 left, and after 10 weeks we've got the whole lot gone. So we have a digital registration of the cold. The cell decides I've had enough cold, and then that's remembered through the subsequent development. So what have we said so far? We've said, well, we've got FLC, all of the files expressing FLC. This gradual decrease is because each cell's deciding whether to switch off. And that's actually relatively infrequent, and so it takes a long time for the whole plant to switch FLC off. And of course it's good that it's a long time. The plant needs to have the whole of winter and not be confused by a sort of short cold spell in autumn. It then remains epigenetically silent, and then it's switched back on again. And last year we actually found that mutants that do this are basically enzymes that take away this same modification. So we really see it as a switch. You're on, you switch off, you stay off, and then you switch back on again in the embryo. Now, so far I've only talked about one cold step, and that's this induction of one of those PhD proteins. And that's important for this cold-induced nucleation of this red modification at this internal site. And that fits in quite nicely with this slow switch, because actually it's activated very slowly in the cold. This is this protein VIN3. So we thought, is that the only cold step? So then we looked in a VIN3 mutant, and in fact the memory is gone, the gene switches back on again, but all of the cold reductions seemed to be perfectly fine, suggesting there were going to be additional cold steps involved. So that took us into the wonderful world of antisense transcripts and non-coding RNA. And at the time, my lab were very worried about my penchant for RNAs. But, this was in 2009. But, at the moment, suddenly, non-coding RNAs are everywhere in everything. And there's huge rows about how important they are. So, it's an interesting paradigm. I think non-coding RNAs are very central to chromatin regulation generally. Anyway, what we had found was that, so here's our schematic of our gene again. Here's this long intron, and these are the exons. And what was quite interesting was there's a little patch of heterochromatin right at the end of the gene. Now, when we published this was very strange. Heterochromatin was normally in centromeric repeats or transposons in the genome, especially in the Arabidopsis genome, not in developmentally regulated genes. But this little patch of heterochromatin with small RNAs was actually turned out to be the promoter of these antisense transcripts. So they start pretty much adjacent to the sense poly-A site, and they go the whole way across the gene, and the most distal polydenylated one is upstream of the transcription start site of the sense strand. So they totally encompass the sense transcription unit. And they're alternatively spliced and they're alternatively polydenylated. And they get very complicated, and you'll be very glad to know I'm not going to try and explain all of this. We've published a lot of this, but the splicing and the polydenylation are key parts of what's going on in this chromatin regulation. But what I'm going to do is just show you one piece of data where we tried to figure out, well, just how important are they for vernalization? And we did it in a fairly simple way. This is the wild-type gene with its promoter here of the antisense transcripts, which my postdoc called cool air. He thought it was hilarious at the time because there was hot air in the mammalian Hox silencing. And so anyway, we now have to live with cool air. But it's amazing, people do remember this name instead of sort of X2, 2G, blah, blah, blah. So it's okay. So cool air is the set of antisense transcripts. So this is the endogenous locus making the antisense transcripts, because we wanted to take away that promoter, but it's part of the 3' end of the sense transcript. So we replaced that with the 3' end of actually of a Rubisco gene so that it didn't make antisense, but we could finish the end of the sense gene properly, and then made pools of transgenics, and then looked at the reduction of FLC in the cold. This is a time course with increasing weeks of cold. Here's our wild type with pools of transgenics, and here is the expression of the transgene now lacking the cool air transcripts. So it certainly attenuates the downregulation of the FLC transcripts. So yes, we think this actually is an important first step in the vernalization process. So let me just wrap up. I'm now going to just summarize what I've said so far, and then I'm just going to finish off by talking about adaptation. So here's our time life cycle. We've got autumn, this very complex arrangement of alternative processing of the antisense transcript sets the expression state. FLC is high. The plants begin to feel the cold. These antisense transcripts transiently accumulate, and they are transient, and that helps shift what's going on, together with accumulation of this PhD protein, which also shifts now to give you a switch from your green to red state at this internal site in the gene. And the cell has decided in a cell-autonomous way, yes, I have had cold. And that happens with all the cells slowly through the length of winter. So then epigenetically remains off through many cell divisions, and then it's switched back on again in the embryo. And as I say, we found that that's involved a K27 demethylase. So it's something that takes away the red mark. So that made a lot of sense for this switching mechanism. So I'm just going to finish off with adaptation, because it's I think such a nice story this now, and I can show you some nice pictures of where I get to go and visit every summer. We started this many years ago because actually, when you think about it, winter is very different indeed in different parts of the world, and Arabidopsis thaliana has adapted to pretty much from the Cape Verde Islands all the way up to the Arctic Circle, the same species. So how does it do that? And we collected over the years many different natural variant types from different places, and have characterized their flowering time and their vernalization response, and there is really extensive natural variation. Now, when we go in and look at FLC in and of course the whole genome sequencing is helping us do this, but we ended up sequencing, I think it was 85 on here, FLC types. We just said, well, you know, do they all look the same? Are the amino acids different? What's going on? Turns out that actually they fall into different groups. The amino acid sequence of all of these is identical for all the FLCs. We've done a lot of the QTL analysis, which I'm not going to get into, which actually showed FLC as a major QTL underlying both flowering time and vernalization response. So we were looking for what the molecular variation was, and it was clearly not at the amino acid level, the protein function level. And so these different, if you put it on this splits tree, you see these different groups, which are actually very similar to each other. Not all necessarily geographically kind of linked, but they're clearly different haplotypes. And they've followed the different types. So some of them we call RV, they're rapidly vernalized accessions, so they only maybe need 4 weeks cold. They will tend to be from the south. But they're not all, so you get rapid and slow vernalizers, the vernalizers in both north and south. These was a slow vernalizer and so on. So we have these different groups defined by SNPs in the non-coding regions. And what's emerged over the last couple of years is that each of these have arisen from actually just a small number of SNPs, and they confer a very different regulatory mechanism onto FLC. Well, very different. They just modulate what's going on. The first one, and this has just got published, I think was extraordinary to me. This is one of the ones, this is SV3, so it's this clade here, where one of them is defined, a lot of these are in northern Sweden. You can sort of see the names of the countries here. We've determined that actually it's a single nucleotide in the intron of the antisense transcript. It's actually also in the intron of the sense transcript, this one here, which affects the splicing of the antisense. And through that, it affects the level of transcription of the sense transcript. A single nucleotide. It's actually next door to the end of the dinucleotide of the intron sequence. But you know, when you're looking at genome sequence, you know, most people sort of ignore intronic variation. And yet here, a single SNP, and this has just come out so you can read it, we did all the complementation, etc., defines that Swedish haplotype. So really very little change is required to give you an FLC which is actually much more highly expressed non-V and has a slower vernalization. The other one we looked at in detail was this one from Lervik, northern Sweden. It was extraordinary in that this is now looking at the acceleration of flowering, so leaf number is our assay, with increasing weeks of cold. And you can see some of these need really long cold, 12 weeks before you have a good acceleration of flowering. So this orange one, you gave it 4 weeks cold, and it barely noticed it had had any cold. That was, you know, what was remarkable. So we followed up Lervik, and Lervik is actually one of these that it's from really right up to the sort of northern limit of the Arabidopsis range. Now, when we look again, this in autumn. We have high FLC. It goes off in the cold and then stays off. So this is a rapid vernalizer, typical of Frey-Cole or any of the ones that only need 4 weeks. When we looked at the Lervik in that scenario, what happened was the gene was repressed quite happily during the cold. It then just got switched back on. It was a bit like a vernalization mutant. But if you gave it 12 weeks cold, it vernalized just fine. So all the trans factors there were fine. It just took longer to actually switch to the epigenetically silent state, perhaps fitting with the fact that it came from a very much longer, a place with a very much longer winter. And then when you look at this nucleation, drawn slightly differently here, but we're looking again at accumulation of our red mark at this internal site. Here's how fast it takes. It takes 4 weeks to get pretty much maximum level of accumulation in our rapid vernalizer. But in our Lervik accession, it took very much longer to accumulate the right level of this red mark to actually switch the gene off. So something was just making it very much slower accumulation of epigenetic silencing. And when we map the determinants of that, and this is now the FLC of Colombia and the SNPs in this Lervik accession, we can map that determinant to just 4 SNPs, again, 4 non-coding SNPs in that region where actually all of these trans factors bind. And that makes the difference between needing 4 weeks cold and 12 weeks cold. So really, we still don't absolutely know yet how those 4 SNPs work, and we're carrying on looking at that. So it just showed us the power of actually really mining in on accessions adapted to these different places, and to try and really understand the basis of the adaptation and how actually the plants are monitoring this very complex temperature profile. Because you remember, when we vernalize in the cold room, it's a nice constant temperature, but out in the field, the temperature's going up and down all over the place. So we've got these little red dots here are the sort of Google Maps, all the accessions we've collected from Sweden. And here's Lervik up here, right on the Arctic Circle. And we've got a big project to understand, how are plants integrating this fluctuating temperature over these long time scales, and why do some accessions manage to do it in 4 weeks and others need 12 weeks? So this is Lervik, wonderful place. I've been twice so far, and it's always been sunny, so I have this wonderful vision and memory of Lervik. It's a beautiful area up there. This is Svante Holm, who actually, it's been wonderful. He works at the University of Mid-Sweden, and it's fantastic to have a collaborator who lives nearby your field site. So he's a major part of this. And so we've been planting plants there, different genotypes, and then a poor MS student of Svante, and this is now Lervik in sort of last February, slightly less ideal, has to go and dig out the trays out of 3 feet of snow. She digs them out, harvests them, and sends the material back to Norwich so we can do the analysis. So we have this time series experiment going on to look at FLC silencing in a natural winter. They look remarkably fine, these plants, given what they're being exposed to. But what's really interesting, so here I was saying to you the really fluctuating temperature. This is some loggers that we've got on the ground at this site in Lervik. The temperature's going up and down, and you can see in July it goes really, because it's at the low angle of the sun, it gets really very hot and then very cold. So somehow the plant is integrating this very complex temperature. This is obviously when the snow is on the ground, the temperature becomes much less variable, except when it's blown away. And one thing that was surprising to us was we had assumed this is when vernalization would happen. But it turns out that absolutely, this is way too cold for vernalization. The plants need to be at 0 and above to vernalize. So there's no vernalization here. And in fact, in Lervik, the plants are fully vernalized by the end of November. And then they just sit and wait for the snow to go and for the days to warm up a little bit. And they flower immediately, the snow's gone. So somehow the plant's integrating this massively fluctuating temperature and registering the vernalization signal. So that's our current challenge, to figure out how that happens. Hugely important process, of course, as our winters are changing, we've got to understand this perception of temperature. And not only how the winters are changing and the temperatures are changing, but of course the temperature change relative to the photoperiod. The photoperiod is not going to change. So this intricate network which has evolved to combine, am I in the right photoperiod and the right temperature, is being disrupted. And that's going to have huge consequences for many crops. And we've got quite a big program to look in actually purple sprouting broccoli, Brassica oleracea, with Jim Monaghan here, and this is my colleague Judith Irwin, to actually look at FLC in the brassicas, how important they are, and how can we actually define haplotypes and alleles which are perhaps slightly less sensitive to some of these fluctuating temperatures. So let me finish off by just giving you a few summaries rather than long lists. There's 3 take-home messages I want you to leave you with. One is, and it's going back to really the importance of the reference plant. I've avoided saying model system. Model sort of suggests that they don't live outside the Arabidopsis. This is a good reference system for ecology as well, so it's more than just a model. And actually, when you understand the trait, a very complex trait like flowering time, if you really understand it, it actually comes down to sort of a relative genetic simplicity. And actually, I think it's important to stay with the reference until you get to that level of understanding before the sort of push that comes inevitably to translate it into various strategic scenarios. It's important to do the translation, but if you do it too early, it'll happen more slowly. The second is, when we look at something like vernalization, which, you might imagine vernalization was the most plant-specific thing you could probably study. You don't find many, I guess, hibernation, but it's a completely different mechanism. It is a similar thing in animals. But it comes down to a very conserved mechanism, and it's a switching mechanism induced by this prolonged cold signal. And the lessons coming out of FLC, the non-coding RNA, the chromatin, are actually very important for some of the systems in the mammalian switching. And lastly, this whole analysis on adaptation, there's been massive variation for natural variation. There's been massive variation to adapt to different winters and growth in different parts of the world. And adaptation, the molecular basis of adaptation, we know very little about. And certainly the story and the message that's coming out of FLC is that it can be relatively few SNPs having quite big phenotypic changes. And that's quite encouraging in terms of adaptation in a climate change, because it won't take so long for 1 or 2 SNPs to accumulate. So on that, I just want to thank my group, the funders, BBSRC, who've supported me for a long time, ERC more recently. This is my group. I haven't gone into this, but we have an active collaboration with Martin Howard, a computational biologist who's at the John Innes, and he and I have a sort of, occasionally a sort of lab day out. So they made me go to Thetford Forest and swing through trees last summer. So this was fortunately a nice sunny day doing that. But this is the group who's currently doing the work I've talked about. And I thank you for your attention.